And when these junctions are actually weak, you can see like these peptides can sneak through these membrane, and it will be engulfed by the antigen presenting cells, which present them as a epitope on them, and then it triggers like a whole cascade of the immunological reactions, and then it will lead to the removal of the microvilli from the intestinal lining, as you are seeing here. So normal intestine, we have these villi, and in a abnormal intestine where we have the celiac disease, this brush border is completely removed, and it actually causes to the malnutrition. So the right now the solution to this problem is like go for a abstinent diet, so we can quit eating on gluten. This seems quite simple and a straightforward approach, but it has a lot of complications. You can imagine like a country like India, leaving wheat is not that easy. Another problem is like when we actually avoid eating wheat, it actually deprives us for the many nutrients, like especially the fibers, because wheat is also loaded with fibers which are required for maintaining the gut health. And I think the previous seminar that you guys have heard, you have heard about the importance of the gut health and how it actually influences our mood swings and many other things. So the gut health is extremely important, as we learned, and it could be actually deteriorated by actually leaving these grains which are loaded with fibers. And these fibers are actually fermented by the microbiota in our gut to stay healthy. Another problem that happens is like when we go on like these gluten free diets, they are loaded with carbohydrates. So it leads to the increase in our body mass index and also deprives us of some micronutrients that are rich in wheat grains. So we thought like we can actually to find some solutions through the crop biotechnology. And these solutions are either we can eliminate gluten or we can detoxify gluten. So I will first talk about the gluten elimination. So in this approach, there are two different kind of promoters that regulate the expression of these gluten proteins in wheat grains. So some of the promoters, they are actually methylated in the vegetative tissues, as has been shown. So these proteins are they does not code until these methyl groups are removed from the promoter, so to promote their transcription and followed by their translation in the seeds. The other kind of the promoters are these which are protected for methylation, but they require induction of tissue specific transcription factors. So interestingly enough, the promoters of the gliadins and low molecular weight glutenins, which are the major causal agents of the celiac disease or are loaded with the epitopes that lead to the celiac disease induction, are actually such kind which are having the methyl groups. The other kind of the gluten proteins, which are called the high molecular weight glutenins, their promoters are actually protected by for the methylation, and they can only transcribe when the tissue specific transcription factors express. So this is quite interesting because by this means we can specifically eliminate the toxic gluten proteins, because I would like to emphasize here, not all the gluten proteins are bad for our health. The high molecular weight glutenins have very little epitopes, and you will see in the later slides also that I showed like the number of epitopes. And you can see like these proteins are also essential for baking properties. So if we eliminate completely like all the gluten proteins, it will affect the baking properties of the wheat grain. So first, like to move on with this strategy, we decided to clone this gene that removed the methyl groups, and this gene is called Demeter. So we decided to clone this from wheat and barley. Barley is a sister grain to wheat, and it is used largely for beer production, as well as it is used as food in India and also as feed. So as you guys might be aware, the wheat is a hexaploid. That means each gene exists in 6 copies in a diploid organism or 3 copies in a haploid organism. So to clone, we actually adopted a microarray hybridization approach, which is you hybridize the back filters with the cDNA clones of the genes. We identified 3 copies of the gene, we get it sequenced completely, and then we decided to map it to the subgenome. So that I'm showing here on this corner, like where my cursor is, you can see like it maps to three wheat chromosomes called 5A, 5B, and 5D. And we use like a special DNA, which is the DNA of nullisomic and tetrasomic lines. Who have not heard about these stocks? So nullisomic means there is a loss of chromosome and it is compensated by another chromosome. So there are, as I said, like they are homologous chromosomes. So they are not homologous, they are homologous chromosomes. So these homologous chromosomes are not true homologs or copies of each other, but they can actually compensate for the loss of one. So if we delete one chromosome like 5A, and we have the double dose of the 5B chromosome, it can compensate for this loss. So that is what has been done here. Like, so it is nullisomic for 5A, nullisomic for 5B, and nullisomic from 5D. That means the 5A is missing, 5B is missing, 5D is missing in this chromosome. And these DNAs are the DNAs of the ancestral subgenomes. So when the wheat evolved, it actually evolved by the crossing of these diploid grasses like Triticum urartu, Eglops speltoides, and Eglops torsii. So we have access to these DNAs. So we use these DNAs to map these genes to the specific subgenomes. On this end, we were trying to actually establish a connection between the Demeter and the translation of the gluten proteins. So what we did, we collected the samples from 5 days after anthesis. So anthesis means at the time of flowering. So 5 days after flowering, 7 days after flowering, 8 days after flowering, 10 days after flowering, and 13 days after flowering, we collected the samples. Because we want to study like when these gluten proteins start to accumulate and when these this Demeter homologs express, so that we can establish a connection between the expression of Demeter and expression or loading of the gluten proteins into the grain. As you can see here, when we actually, this is the transcript profile of the Demeter genes. You can see like at the 5, 7, and 8 day post anthesis, especially at the 5 and 8 day post anthesis, all three homologous copies of the gene express almost equally. But at the 7, 8, and 10 day, like you can see, otherwise the A subgenome, which is shown in red bars, and the D subgenome, which is shown in the purple bars, they show the opposite expression pattern. You can see like when the expression pattern of A rises, the expression pattern of the D decreases. But the expression pattern of the B genome copy stays stable up to 10 day post anthesis. So we figured out for triggering the expression of the gamma gliadins, which express at the 8 day post anthesis, it is important to have like the induction of the D genome and B genome, because you can see D genome is expressing very high at the 7 day post anthesis. And you can see here the gamma genome start to express at the 6th day. And here also you can see in this profile like where the accumulation of these proteins have been shown. So you can see like at the 8 day post anthesis, which is shown in the red color, it's the gamma genome peaks is started to appear. You can see here in these peaks where my cursor is at the bottom. So that establishes a connection between the expression of the Demeter B genome copy and Demeter D genome copy and the expression of the gamma gliadins. At the 8 and 10 day post anthesis, you can see the expression of the alpha, the 5A copy start to appear and increase in number. And then at the same time, the expression of the alpha gliadin and beta gliadin start to appear. So that means for the induction of the alpha beta gliadins and low molecular weight glutenins, which is shown in blue here, you can see like this is the alpha beta gliadins, which started to pile up at the 10 day post anthesis. You can see like the emergence of the green peaks. It has started to appear in here like at the 10th day, and then by the 13th day post anthesis, the expression of the D genome copy expressed highly, and that is correlated with like the expression of the remaining glutenins, especially the low molecular weight glutenins. So you can see like the function of these Demeter genes is subfunctionalized because they are not doing the same function, rather they somewhat subfunctionalized. So they have decided what each gene will do, and then it induces the expression of one sort of gliadins and glutenins. So now to validate uh, our hypothesis, because we said like the expression of Demeter is correlated or connected with the expression of gliadins and glutenins, we decided to screen for the mutation in the Demeter genes. So for this purpose, we actually decided to screen the tilling libraries in two different wheats, Kronos and Express. So Kronos is a tetraploid wheat. You might have heard about the pasta wheat. So pasta wheat is a tetraploid. That means it has two copies of the gene. The Express, which is a hexaploid wheat or bread wheat, it has three copies of the same gene. We screened these libraries. We identified in Kronos a total of 77 mutants, and in Express we identified 149 mutants. You can see like we only chose to study the active site of the gene for the mutations. The reason was this gene is very large, about like 5 kbs in size. So it will be a lot of effort to identify mutations in the whole gene. And it is, we hypothesized that it is really important to actually look at the active site of the gene. If we can find mutations there, we can disrupt the function of this gene. So we identified these single mutants in A, B, and T copies of the genome, and then we looked the protein profile of these. These single mutants, as you can see here, they showed like a elimination of a specific prolamins or these gluten proteins, like some having like deletions in the gamma or reduced expression of the gamma genome, and some has lacking the omega gliadins. But none of these will be actually completely devoid of the glutenins or gliadins. You can imagine like when you are only deleting one copy of the gene, so there will be still two intact copies. To deal with this problem, we thought like we can cross them together by cross fertilization. But when we crossed, we never found a double mutant. You can see like you might see this one, which is a double mutant, but like this is a double mutant of two substitutions in the genome. But when we try to cross, like where we have a stop codon, a premature stop codon, or a splice site variant, we were not able to get a double mutant. To find the reason behind it, we studied the anther morphology because we suspected its role in the anther development as well. When we studied the anther morphology, you can see the size of the locules of the anthers got reduced by knocking down this gene, and also the number of viable pollen. You can see we use the Alexander staining to study the pollen viability, and if you see a dull color pollen, that means like the pollen is not viable. So we identified like it affect the anther locule size, it affect the number of viable pollens, and it also affect the pollen germination. So it has a vital role, and that explained why we were unable to get a double mutant in a tetraploid and a triple mutant in a hexaploid, because the plant cannot afford to have all the three copies removed. So to deal with this, before that, like I will also talk you like as I told you earlier, like we also tried to clone this gene from the barley. We thought like we can also induce mutations in the barley Demeter gene. So for inducing mutations in the barley, we took a very novel approach. We took like this Xentomonas bacterium. So this bacterium naturally causes two diseases. One is called the bacterial leaf streak, as you can see here. And then another disease it causes called pseudo black chaff, that turns the chaff of the spike into black. So this bacterium has a type 3 secretion system, as you can see here. So it can inject its own proteins. At the same time, if we can actually transform this bacterium with the engineered protein, it can also deliver that to the plant cell. So that's what exactly we did. We actually modified one of its tail effector proteins to target the Demeter gene. So this tail nuclease is an artificial protein which is attached with a moiety of the fork1 nuclease, attached with a domain which can recognize a site in the Demeter gene. So it reached into the hope was it will reach into the DNA of barley and then can create mutation at the site that we decided it to bind. We loaded the spikes at this stage, like which is called like the boot stage. So we can cut open the boots and then fill it with the bacterial suspension, which is transformed bacterium. And then these bacteria will get into the developing grains, and hopefully while the grains are developing, they go through the meiosis cycle, and it can induce mutations. So what we did, we collected these infected grains, as I'm pointing here, and then we screened 114 of these for the mutations at the target site. and we were fortunate enough to find like 5 plants that were actually having mutations at the target site, as you're seeing in one of these. But the downside here was like when we actually grew these plants, they were completely sterile. So that actually proved the same thing that we observed in wheat. These genes are important for pollen viability as well. So to deal with this problem, we actually decided to suppress this gene in a tissue-specific fashion. So to do that, so by this, I mean like we will only knock it out into the grain, not in the pollen grain. So it will not affect the pollen viability. So we developed a 938 base pair hairpin, where there is a stem of about 180 nucleotides, and that stem targets specifically the Demeter transcript. When we introduced that with the biolistics approach or the gene gun, we identified about 118 putative transformants. We screened those transgenic lines, and 7 of these lines actually have introgression of this hairpin that we introduced. This hairpin was driven, as you can see, with the high molecular weight gluten promoter, so it will only express in the grains. You have seen that in the previous slides. I have shown these promoters express in the grains. So we identified these 7 lines showed reduction in the prolamin content or the gluten content, as you can see, like different lines. So first line is the control, and the rest of these lines are the transgenic lines, which were actually each showing variable pattern of the gluten protein accumulation, as you can see. And this is the HPLC profile of the same proteins, and you can see 2 of these transgenic lines was significantly reduced in the content of these gluten proteins. And this panel is showing the seed morphology is not altered. So some of these lines have showed up to 76% reduction in the content of gliadins. We went ahead with this approach and then created artificial microRNAs. And you might wonder why we need, when we have actually hairpins expressing quite well, and then we suppressed and created the transgenic lines. These hairpins are quite large in size. As I said, like the stem of these hairpin is about 180 nucleotide. You can imagine like we cannot control the splicing of it. It is spliced in the wheat genome automatically into 21 nucleotide segments. And then when this splicing is happening, it could actually create off-targeting of genes. That means it could target not only the desired gene, but some off-target genes. To avoid this problem, we created the artificial microRNAs. The artificial microRNAs are designed from the natural microRNAs. So we identified 3 sites in the Demeter gene, 2 from the N-terminal region, 2 from the active site and 1 from the N-terminal region. And we assembled that on the rice microRNA 528 as a template and provided it with the high molecular weight gluten promoter, as we did in the hairpin, and introduced that into the wheat genome. And we identified about 218 putative transformants. About 39 of these lines actually showed the integration, and 12 of these lines showed significant reduction in the gluten content, as you are seeing here. So the first one is a control where we only introduced the bar gene as a transgene, and this bar is a marker gene. So it is for herbicide tolerance. So it has nothing to do with the gluten accumulation. So it has been used as a control here. And you can see these transgenic lines. And 2 of my favorite lines are actually shown in red, which showed significant reduction in the gluten content. We identified some lines which were actually having up to 89% reduction in the gliadin content. These lines also showed some interesting characteristics, like they have increased lysine content, increased gluten strength. So when you remove the gliadins, it increased the strength of the gluten. So we started with a soft white wheat, which is generally used for cookie making, and we end up getting a wheat dough, which was good for making breads. So this is another gene which can actually help in the same process. So this is called the DRE2. So de-repressed for ribosomal protein S14 expression. This protein is known to actually mount a iron sulfur cluster onto the Demeter enzyme. So same enzyme we just talked about. So this enzyme put like this iron sulfur cluster and make this as a holo enzyme. You might have heard about the APO enzymes, and then it requires the factors to become a holo enzyme. So this enzyme does that function. So knowing like its function, we decided to actually create mutations in this gene and then study the effect of creating mutations in this gene on the prolamin accumulation or the gluten accumulation. So first, we decided to clone this gene from the wheat genome, as we did for the Demeter. So we in small pieces in segments, we cloned this gene first, and then we decided to map this gene. It mapped also on the 3 homologous chromosomes. This time, the chromosome 2AL, 2BL, and 2DL. So L means the long arm of the chromosome. And then we also studied the expression of the 3 copies of the gene in the wheat grains. And this Z is indicating of like the developmental stages of the spikes. And then we studied like the expression, and we noticed like the 2AL copy strongly expresses in all developmental stages over the B and D copy. So we screened the Chronos stealing population as we did in the Demeter's case. Like there we screened 2 populations. Here we only screened the Chronos population, which is tetraploid wheat, and easy to handle because it has only 2 copies of the gene. We identified mutations in the, you can see the premature stop codons in the A genome copy and a splice site variant in the B genome copy. And these are the protein profiles of these. The same with the Demeter thing, like you can see some elimination of proteins, but does not see a complete loss of these proteins, um, which is explainable. As you know, like earlier we talked like we need to cross them together. So we did the same thing, we tried to cross them together, but the same effect we noticed, like when we tried to cross, it was actually not giving us any double mutant. So to deal with this issue, we decided to switch the approaches. We decided to use the site-directed mutagenesis approach. And you guys might have heard about the CRISPR technology. So we actually adapted to this technology. To take full benefit of this approach, we decided to create some insertional mutations into the DRE2 gene. So we were trying to insert a segment, which is the Demeter tail repressor. We have just talked about the tails, the tail protein, which are those proteins which can specific, which could be guided to specific sites in the genome. We, rather than attaching it with a nuclease domain, as we did earlier in the Barley's case, we attached it with a repressor domain. Repressor domain is opposite of the transcription factor. Instead of inducing, it repress the transcription. So we thought like we can introduce this piece of the DNA into the DRE2 and then can have the double benefit. Like, so this repressor can repress the Demeter at the transcriptional level, and this fracturing of the DRE2 gene can actually affect the Demeter at the post-translational level. As I told, like it installs the iron sulfur cluster, so it will avoid it making into an active enzyme. But after introducing this by the bialistic or the gene gun approach, we identified 100 some transformants, and we screened them for their protein profiles. Some of them showed reduced accumulation of prolamins. But I will tell you, like when we screened them at the genomic DNA level, none of the mutations, none of these lines actually carried an insertional mutation in the DRE2. So what we found is like the transgenic lines where this piece was individually introgressed, but we could not find where we have an insertional mutation, which is also kind of explainable because the homologous recombination based insertions are very rare to identify. So we took another approach. We decided to actually silence these gliadins and low molecular weight glutenins directly by introducing a chimeric hairpin construct. So in here, like the earlier hairpin, the stem is actually chimeric. It was carrying the pieces which were complementary to different sort of gliadins and low molecular weight glutenins. And we assembled them together, so each one of them can actually knock out a specific group of proteins. And this is just showing like the wheat TARC14. It is a kinase gene in the wheat, and it is kind of benign in this purpose. So we took like its intron 14 to make a loop. We attached this hairpin with the gamma subgenome of the Barley streak mosaic virus to be used in virus-induced gene silencing. So this is a transient expression of this hairpin through a virus into the wheat. And then this is done like through the stable transformations. We attached this hairpin with a high molecular weight gluten promoter, as we did earlier, and introduced that into the wheat by the bialistics approach. We identified the same pattern. We saw like the reduction in the gliadin content in the BSMB VIGs, as well as in the transgenic lines. You can see the complete elimination of certain groups, but none of the lines that we identified were actually completely devoid of these proteins. So to move on, like with the technology, we thought like since we have yet not identified a line which has completely eliminated these toxic proteins, we adopted the CRISPR CPF based approach. CPF1 is a variant of the Cas9 protein. It is called Cas12a. So by the time like when we were doing this research, the technology has advanced, and there were procedures developed by Dr. Jian Kangzu of Purdue University here, where you can actually knock out several genes together with this CRISPR CPF based approach. So we adapted this technology, and in this here, like I'm showing how complex this structure of these genes are. These genes are coded by exist on the three group 1 chromosomes and three group 6 chromosomes. So I just showed like one bar, but they exist on 6A, 6B, and 6T chromosomes. And each block that I'm showing here, they're tandemly duplicated. So there are copies, the paralogous copies, and there are homologous copies. So there were duplications within a segment, and there are duplications in between the homologous chromosomes. So we pulled out the sequences of these genes as we did earlier also for the hairpin construct, and we tried to identify the conserved sites. So we identified those conserved regions and then developed three target sites for each gene to knock out with this CRISPR CPF. So if you have heard about the CRISPR CPF, CPF is a nuclease which need to be guided to a specific site. So these guide RNAs guide it to a site. So these guide RNAs, we developed 3 per group of gliadins. And here I'm showing, like as I earlier said, the gliadins are loaded with the toxic epitopes. So epitopes are the segments that the antigen presenting cells that I showed in the second slide actually present and create like a immune response. And you can see here the glutenins, especially the high molecular weight glutenins, which are required for baking properties, are actually very, very low in those toxic epitopes. And these epitopes are not the major epitopes. That means the major epitopes which are very common in the people. The minor epitopes are ones that are very, very rare in the population, the human population. So we targeted these. We identified these targets as I'm showing here, and then we in vitro tested these targets. So you can test these targets beforehand before introducing into the plant in the test tube. So we did the same thing. We actually amplified a segment of the genome. We actually created a nucleoprotein complex, which is the CPF1 enzyme, which is Cas12a, and the guide RNA, and we mixed them together. And then the guide RNAs that actually performed nicely or digested this product were selected to create this plasmid, where you can see we developed the array of guide RNAs. So this special plasmid has been developed by a scientist in the Maryland University here. So we obtained these resources and modified it, and we installed like these guide RNAs for alpha, gamma, omega gliadins and the low molecular weight glutenins. So this complex structure, this part is actually creating or having a complex for the guide RNAs. This part is actually expressing the CPF1 enzyme, and the promoter is a maize ubiquitin promoter. So this is the homologous recombination between 3 different plasmids that give rise to this very complicated plasmid with 3 expression cassettes. So first expression cassette, as I said, is maize ubiquitin promoter attached with the CPF1 enzyme and nopalin synthase terminator. So each gene has required a terminator to terminate the transcription. And then next cassette contain another maize ubiquitin promoter, and then this guide RNA module, and then it is attached with the nopalin synthase terminator. And the third expression cassette is the 35S promoter, hygromycin resistance gene, and then it's the terminator, the poly-A terminator from cauliflower mosaic virus. So we got this plasmid created, and then we introduced that by the 2 different methods. One is the gene gun or the biolistics method. The other one is the Pollen magnetofaction, and I will describe that in the next slide. This method, which is where the carbon nanotubes could be used as a delivery vehicle, is yet to be perfected. So we are working on this. So this is a new method of delivering genes or DNA into the plants. So pollen magnetofaction, here we use the magnetic nanoparticles. So these are iron oxide nanoparticles, which are 200 nanometers in size, and it could be coated with the DNA and then delivered using a magnetic force into the microspores of pollen grains. So I'm showing here the workflow that we optimize for wheat. So one can collect the wheat spikes at the Ficki's developmental stage 10 or 10.2 and then expose it to the sunlight or artificial light. It protrude out the anthers as I'm pointing here. You can collect the anthers, then extract the pollens, then magnetofact the pollens on a magnetic plate as has been shown here. Then you can study the pollen viability because one has to dry the pollen after magnetofaction on a filter paper. After the drying, you can study the pollen viability with the Alexander staining, as I have already shown and described. Like you can see the dead pollens are light in color, the bright pollens are living pollens. And then you can count the number of the living pollens using a hemocytometer, as we did here. And then we also studied the pollen germination by doing the germination test. And then after that, like you can actually artificially pollinate the spikes. And here we were spraying it with the gibberellins to induce the seed formation. Then we collected these seeds and studied the seedling if it has the DNA in it. And then the lines that showed like the introduction of the the plasmid that we introduce, we screen them for the digestion or introduction of the mutation. If you have like introduction of a mutation at the target site, you will see digested product. So this is the intact product, this is the wild type control, and this is the transgenic line. So it has only mutation in the gliadin gene, not in the gamma gliadin gene, because you can see there is no digestion product here. So not only we screened like we use these transgenic approaches, we also tried to find mutations, other mutations. For this purpose, we looked for the ABA insensitive and hypersensitive mutants. And one might think, why abscisic acid mutants? So there is a mutant in barley which is very high lysine and reduced in the gluten content, which is ABA insensitive. We thought like we can also follow this lead and then identify mutations in wheat. So we screened several mutations in different backgrounds, like in scarlet, which is a hard red spring wheat, and then brewer, which is a soft white winter wheat. So soft white wheats are used for cookie making, hard reds are used for bread making. So we screened mutations. We found interesting mutations which were lacking several gliadins. You can see like this is the control, and then the control is shown in red here in this HPLC profile, and then these are the mutants. So they were significantly reduced in the amount of glutenins. So our hypothesis was correct. So wheat also follow the same pattern as the barley did. So we also identified mutations in the hypersensitive for ABA. So I believe like you understand the insensitive mutants are the mutants in the signaling pathway, and hypersensitive mutants are the mutants which are actually deficient in ABA. So we identified these VARM mutant called VARM1 and VARM6, which were also showing reduced gliadin content. Other than that, we also tried to cross together some omega gliadin mutants to create omega gliadin null mutants. So these mutants were obtained from Italy. It was not created by us. And when we studied the immunoreactivity of these omega gliadin null mutant that we developed by crossing several of the omega gliadin mutants, it showed 35.7% reduction in the immunoreactivity when we studied with the sera of these gluten allergic patients. And these are the examples of the mutants which where the GlyA2 and GlyD2 loci were deleted. So these are alpha gliadin mutants, and these mutants we obtained from Italy. So we were actually now trying to cross this mutant with this mutant to obtain a mutant which will be lacking in omega gliadins and alpha gliadins. You can see here this, when this alpha gliadin mutant was tested by proteomically, it was significantly reduced in the content of epitopes or toxic epitopes. These the GlyA2 mutant, which is not a double mutant, a single mutant, was also tested in an organ system. So you can actually have the organ peels and you can culture them with the gluten protein derived from these lines. That's what has been done here. So wild wheat proteins, when it was fed to the epithelial cells of the celiac patient, you can see it actually blunted the epithelium. But when it was fed with the GlyA2 mutant, it looks very much like the wild type or a healthy intestine thing. Like, so you can imagine when we create a double mutant, we can actually increase the effect and could have like a wheat line which will not affect the epithelium like in this case. So other than that, like as I said, like we also decided to not only eliminate gluten, we also decided to detoxify gluten. Detoxification means we can digest the gluten into small bits. For this purpose, we screened the proteins from the bacteria and also from the barley. And we identified there is a protein called prolyl endopeptidase from Flavobacterium meningosepticum, and then a protein from barley, which is EPB2, the endoprotease B2 from barley. So we took like 4 parameters: substrate specificity, so which protein cuts where. So these endoprotease from barley cuts specifically at the glutamine residues. And these proteins, which are called prolyl endopeptidase, they cut specifically at the proline residues. And as I was telling them, prolamins, these proteins are loaded with glutamine and proline residues. So if you can cut close to these residues, you can actually completely digest these proteins. And I would like to tell here, like, unfortunately, none of our gut enzymes can completely digest these proteins. Substrate length, these proteases, as you can hear, like it is a protease which can cut the proteins. This is a peptidase. This can only cut the peptides. So it creates the template to cut by this peptidase. So protease cut first, and then it is cut by the endopeptidase, and it fits with the pH optima of these enzymes. So based on all of these parameters, we selected these proteins. The pH optima means this work at a pH of 4 to 7, and this works at the pH of 7. So this is the pH in the stomach, and this is the pH in the small intestine. So this fits perfectly with our digestive system. So we introduce these two enzymes into the wheat grains. Attached with the same high molecular weight gluten promoters, identified the transgenic lines, and you can see that control is shown in the blue. B96 is the Brindage 96 wheat variety that we transformed, and this is showing like the transgenic lines. So transgenic lines are showing significant reduction in the amount of these toxic epitopes. So now we are talking about the epitopes, not the protein quantity that as we studied earlier. So as we want to introduce these enzymes and bake these breads, these grains into breads, we decided to increase the thermostability of these enzymes, because you can imagine, like, if you introduce these enzymes and these are being baked, they will the baking process will melt these enzymes. So we decided to engineer the enzymes that can survive the heat. So we modified these enzymes using two different approaches. This is site saturation mutagenesis approach that we followed to modify the prolyl endopeptidase from Flavobacterium. So we introduced several mutations, and mutations only at the four sites, 412, 413, 14, and 15 amino acid residue, actually created a mutation that can survive up to 90 degrees Celsius. As you can see, the mutant number 5 showed, mutant number 10 showed the activity, which is above about 50% of the activity of the natural enzyme at the 90 degrees Celsius. So we heated this enzyme for 15 minutes at 90 degrees Celsius, and it still retained activity. And we are talking about the naked enzyme, like when the enzyme is protected in the grain, it can survive even better. So we also engineered the other enzyme called EPB2, and here we adopted a different approach. We took like a vitamin C as a reference, which is an enzyme very similar to EPB2, and it can survive up to 70 degrees. So we modified these specific residues that I circled here into this EPB2, and we obtained a line or a enzyme which can survive up to 60 degrees Celsius, which is not much because we want it to survive up to 90 degrees Celsius, which is the baking temperature in the core of the bread. But it is a nice indication, like one can create such sort of enzyme lines. So we are still working in this area. So other allergies that I would like to talk is the peanut allergies, and the peanut allergies are on rise in United States. As you can see, in 1997, there were only 0.4% of the US population that is affected, and by 2008, it is 1.4% of the population that is affected. And there could be several reasons behind this increase. Like, I mean, the people are more and more reliant on the fast foods these days. And the fast food, the problem is it is finely processed foods, like which have the loads of these proteins just to create the nutritional aspects of it. So in peanuts, there were actually 16 proteins identified, and these are called ARA proteins in peanuts. So out of these 16, there are 4 proteins which are the major immunogens, like ARA1, ARA2, ARA3, and ARA6. So the solution is same, like either you stop eating on peanuts, but as you know, like in India and Africa, like in many other countries, the peanut is a great source of proteins, and it is also cheap to obtain. So we decided to engineer or identify hypoallergenic peanut lines. So to do that, we first start to screen the USDA mini core collection. So mini core collections are kind of the gist of the diversity available in the in a collection. So in the United States, we have a collection of about 10,000 lines. So people have reduced the or eliminated the redundant lines and then identified the unique lines, which kind of is the gist of the diversity. So there are 108 genotypes. We screened them for their protein profiles and identified lines which were actually lacking specific allergenic proteins. As you can see, they were missing specific protein bands. So not only we screened the USDA collection, we also screened a collection from India, like which is available in ICRI set Patancheru in Hyderabad. So we obtained this collection of 184 lines, which is also representing 14,000 lines diversity of these. And these lines were collected from 92 countries. So this is a very diverse collection. We screened this collection for the same way, and we identified about 10 lines which were actually having reduced content of the allergenic protein. As you can see in the densitometric profile, this is a control line, and this you can see like some of these lines which were actually having significantly reduced content of these toxic proteins. So here, I mean, this is a very busy slide. I'm sorry about that, but I will explain it one by one. Like, so in the first part, what we converted these protein profiles into the digital data. Like, I mean, each a presence of band is indicated as 1 and the absence of band is as 0. And if the band is light or dull in shade, it was indicated in a yellow color. And we changed these into digital metrics so that we can identify lines which could complement each other and could be crossed together to get a line which was completely lacking these four toxic proteins. So not only we screened these lines for the proteins, we also screened these lines for the FED3A, FED2A, and FED2B genes. This is a enzyme which is kind of convert the oleic acid into linolenic acid. So if you stop the conversion, you can increase the content of the oleic acid, and that increase the shelf life of the oil. It makes the oil very healthy. So fortunately, we identified 2 lines which were carrying the natural mutations. So there were 6 lines in total which were carrying the natural mutations and natural double mutations. So both A genome and B genome copy. So the people who don't know, like on the peanut is also a tetraploid. I should have mentioned that. So it has also 2 copies of this gene. So we identified these natural mutants, and 2 of these lines are also showing reduced content of those toxic proteins. So that's super interesting. And then here what we did, like as you have seen in the previous slide, there is a genotype called Arachis diagoe, which is a kind of a diploid or a wild peanut, which is actually quite low in these toxic proteins, as you can see here. And the Gregory is a tetraploid or a cultivated peanut. So we crossed the diagoid with Gregory to transfer this phenotype from this diploid to a cultivated species. So this is what we are showing, like the progeny of this cross. And then the idea is actually to identify progeny lines which were actually lacking several proteins. As you can see, there are progenies which are lacking several of these proteins to cross with the other lines that we identified from the Indian and the US collection. Like the wheat, we also decided to go for, genome editing. So we decided to actually induce mutations in these genes. Like, so these are coded by these ARA proteins, as we talked, and they exist in the homologous chromosomes. So 6 is complementary to 16, 8 is complementary to 18, and 9 to 19. So there are 6 copies of these genes. We took the same approach. We aligned these genes, identified the conserved sites, and developed the guide RNAs. And now we are in the process of introducing that into the peanuts. Dr. Matthew, do I have more time, or shall I take the questions at this time point?
No problem, sir. You can carry on.
Okay.
Oh.
So the second target is like to develop the sustainable crops. So for this purpose, I will take several examples in the upcoming slides. So in this project, what we did, we decided to develop the annual cotton genotypes. So as you guys might be aware, the cotton is a perennial plant, and we actually use it as an annual. So we reap annually and then destroy the plant. But what happens? The plant actually is a perennial, as I said, so it keeps the buds. So it does not use all the resources that it has to produce the balls. So since the cotton yields are stagnant from several years, we decided to take a novel approach to change the cotton plant, the growth habit of the cotton plant from perennial to annual. So for that purpose, we screened a core collection, as I talked earlier, of the core collection is the gist of the diversity. So keep that in mind. So we picked like the 44 upland cotton lines, and we screened them for the strong expression alleles of the 5 floral induction and plant architecture related genes. And these 5 genes I marked here. So these are flowering locus T, suppressor of overexpression of constans, leafy, apetla 1, and full or fruitful. So these genes decide like when the plant should transition into flowering and which meristem should turn into a flower or keep it as a bud and develop into a twig in the coming year. So we decided to find like the high expression alleles of these genes in this collection, and we screened it for the two consecutive years for the 3 developmental stages, which is early developmental stages like 10-day, 15-day, and 45-day after the development. So we collected the meristematic tissue and then studied the expression in these lines. So we developed our expression metrics. So this expression matrix is in for each genotype can gain 15 points via this expression matrix. So 1 point for 1 developmental stage and 1 gene. So we have 5 genes in 3 developmental stages, so 15 points. So this the point is granted to a genotype when its expression is more than the mean of the population level expression of a particular gene. So if a gene shows more expression than the mean, then it's granted 1 point. So you can see like the point matrix is here. So we identified these 5 genotypes which were actually having high expression alleles. So S1 is indicating the developmental stage 1, stage 2, stage 3, and these are the 5 genes. So now we are making crosses between these genotypes. So first crosses were already made and they were sent to Costa Rica for increasing. So we will again study the expression of this. Like you can see like some of the phenotypes. So what has happened here, the branch, if you have seen the cotton plant, it actually produces in a sympodial fashion the flowers. So the internodes between the flowers are shrinked. So you can see the flowers come together and they got clustered. So this is kind of a good sign. It is turning into a determinate growth habit. So that's what we want. So all the buds will be consumed into flowers and produce balls instead of actually saving it for next generation. So this is a different project where we actually we tried to develop the biomarkers for heat tolerance. As you guys are aware, the heat is increasing like the global warming scenario. So we need the lines which can tolerate this heat. So we screened 2 soybean genotypes called DS25, which is heat tolerant, and DT97, which is heat sensitive. And we studied the lipid profiles of these lines, as well as we studied the expression pattern of the fatty acid desaturase genes. So when we studied the lipid profile of these after giving the heat treatment, we noticed like the unsaturation index of this DS25 line was actually very high. So that means like the unsaturation level is reduced in this. So less and less proteins were turned into the unsaturated fatty acids. So this actually provide the benefit. The cell, as you are aware, like when the heat is provided, the the unsaturation label automatically reduce. So if you actually have a line which actually could balance the label of unsaturated and saturated fats, it could maintain the cell's volatility. Otherwise, the cell membrane will fuse into from the bilayer to a single layer membrane. So that's why it is important to actually reduce the level of unsaturated fats in this. So we identified this happening into this heat tolerant line when we studied the expression pattern of these fatty acid desaturases, which actually create the unsaturated fats. We noticed the reduction in the content of the fatty acid desaturase 3A and 3B genes. So soybean is also kind of a paleopolyploid. So paleopolyploids are such which look like diploid now, but they were in past were polyploids which now reduce the number of chromosomes and turn into a diploid and behave like a diploid. So it has also the duplicated copies of the genes. So we noticed like both the 3A and 3B copy of the fatty acid desaturase is significantly reduced in the amount, which reduced the amount of the highly unsaturated fats, and it kind of matches with the lipid profile. So we find a biological marker that could be used to breed for this trait or transfer this gene. This is another example. So this is an example where we want to develop lines which can wheat lines which can survive the crown and root rot. So this is a major problem in the United States, especially in the west side of the country and in Australia, like where the root and crown rot actually kills a lot of wheat genotypes. So there are bacteria that can produce the natural antibiotics, which is called pyrrolnitrine. So these pseudomonas produce those antibiotics. So these antibiotics are produced by the concerted action of the four genes in a pyrrolnitrine operon, which is called PRN operon. So these are these four genes which convert the tryptophan, which is a natural amino acid existing in all genotypes, even in wheat, and then could convert that into pyrrolnitrine. So we thought like if we install this operon into wheat roots, it can convert the tryptophan into pyrrolnitrine and can self-sustain. So we introduced we modified the operon first to kind of adapt for the wheat gene. So adaptation for the hexaploid required us to modify the GC content of the genes and then attach these genes with a root specific promoter, because we only want to express this antibiotic in the root, not in the grains. And then we attached, since this tryptophan is only produced in chloroplast, we want to send all of these enzymes to chloroplast. So there are signal peptides, which are like the address you write on the envelope. So it guides the protein to go into the chloroplast. So we attached all of those four proteins that I showed earlier to guide into the chloroplast, and then that can convert the tryptophan into pyrrolnitrine. So here I'm showing like not only we introduced just pyrrolnitrine expression cassette, we also introduced the bar gene, which is a marker gene, as I said, which is used for selection. So here we identified the lines which were actually having introgression for both bar and the pyrrolnitrine. And then when we screened the progeny, you can see like there is an assay called phosphonotriacine acetyltransferase assay, where if you have the bar gene expression, you can see the change in the pH, which is shown which change from red to orange if you have the expression of this. So we identified these lines and then we screened that for the pyrrolnitrine content using the mass spectrometry approach. So triple quadrupole mass spectrometer was used. Here we used like the artificial pyrrolnitrine to actually create a standard curve to determine how much of the pyrrolnitrine is expressed by these transgenic lines, and two of these lines express significant amount of pyrrolnitrine. So now we are testing these lines in the greenhouse. So now I'm actually talking about another approach, like where we were actually developing interesting management strategies to sustainable crop production. So sugarcane aphid is a problem in the sorghum production in sorghum production in United States. So this problem only started very recently from 2013 to 2015. This aphid started to emerge from Texas, and it spread it into about like 17 states now and 400 counties. So counties are like districts in India. So it has spread it very far wide, and there are only two insecticides that are currently being used here to control this aphid. But if you constantly use an insecticide or a fungicide, you develop resistance for it. So we decided to develop a strategy which could counteract the development of resistance. So we have heard about like the RNA interference. So we thought like if we can spray these interfering RNAs onto the plant, it could serve as a control, like a biological control or insecticide. So to decided that, we decided to clone the gene targeted by one of these insecticide called acetylcholine esterase. So acetylcholine is a neuroreceptor in the insect. If you have constant production of the neuroreceptor, it actually kills the insect because too much excitement or like your nervous system is always on alert. So it kills the insect. So we decided to clone this gene. Since there is no sequence available for sugarcane aphid, we took like the cotton aphid sequence as a reference and cloned this gene from the sugarcane aphid, and then identified two sites to develop these double-stranded RNAs. So double-stranded RNAs are the RNAs which have two strands. So the natural RNA just has one, as you are aware. Like it's a so if you have two strands of RNA, it is treated as artificial, and then the cells digest it with the dicer enzyme or argonate proteins, and then it produces like a silencing effect. That's what happened. So we actually fed these insects artificially with this in these cups. So we painted the leaf discs with these artificial double-stranded RNAs and then let the insects feed on it. And after feeding for the 2 days, we actually monitored the deaths in these insects, and we noticed like when we feed a 200 nanogram dose of these artificial microRNAs, significantly reduce the amount of the insects. About 2 and 2.5 fold reduction was observed with both of these. So this indicates like these double-stranded RNAs could be used as a pesticide and can replace the chemical pesticides. The same approach has been used here, like where we actually the Puccinia immaculata is a rust that causes rust in the turf grass. So when we actually have like this rust, it is actually controlled by the PHI, which is a phosphite, is a organic substance which can actually regulate this fungus. But unfortunately, it is not known which genes it target. So we actually studied the expression level of the genes after spraying the phosphite into the fungus, and we identified about 114 genes which showed reduced expression. Out of those, 20 were quite promising. We studied these genes by creating double-stranded RNAs targeting them, and we studied the knockout of those genes. Two of these genes, like glutamate and acetyltransferase and cystathione gamma synthase, showed kind of significant reduction in the rust appearance, as you can see the target 18 and 19. So this again proves like these double-stranded RNAs could actually serve as a pesticide or a fungicide. This is another example where we actually introduce these double-stranded RNAs into the bacterial cell, because as you are aware, the dsRNAs are very labile. They actually degrade in the environment. To have a longer effect, we decided to actually introduce that into the bacterial cells, which can express these double-stranded RNAs. So there are special bacterial strains of E. coli which are actually deficient in RNase reactivity. So you can actually produce the double-stranded RNAs. If you have the active RNase 3, it will degrade the double-stranded RNA. So we transformed these bacteria, and then we actually co-cultured the fungus with the bacterium, and then we monitored the radial mycelial growth of the fungus. We noticed when these bacteria secrete these double-stranded RNAs, it significantly checks the growth of the fungus. As you can see, 10 to the power 7 dose of the cells of the bacteria reduced it as good as like the fungicide. The same we studied with two different fungi. So one fungus caused gray mold in peach, and another one called the root rot in Armillaria is the name of the fungus. It caused the root rot in peach, and you can see also the reduction in its size. So not only this, like we took another approach where we introduced the FANA modified oligonucleotides. So FANA are kind of the DNA or the the nucleotides or nucleosides, which are actually decorated with special arabinose moieties. So this create artificial nucleosides. When you introduce into a oligonucleotide, it makes them very, very stable and does not degrade. And it actually binds to the RNA very, very stably. So what we did, like we identified sites in the genome, the gene, which is called cytochrome P450. And this enzyme we know is very important for the cell wall formation in this fungus Armillaria malia that I showed earlier too. So we identified these targets, and we created the FANA nucleotides. And then when we actually spread this on the plates like the antibiotic, and then seeded with the fungus, we saw like the significant reduction in the mycelial growth with both of these targets that we identified. So with this, in this slide, I'm just trying to show some of the books that I published, and then also summarize some of the research that I did and talked in this. If you guys are interested, you can actually read these. And I would like to thank my group, both at the Washington State University and the Clemson University, and the funding agencies. With that, I will be happy to receive questions.
Thank you, sir, for the nice presentation. Good slides. We really congratulate your works, and your presentation was very nice.
Thank
On
you.
behalf of all teachers and students gathered here, we express our sincere thanks to you, sir. Before going to the discussion part, the resource person and faculty members from Botany and Zoology and student coordinators, please switch on your videos to take a photograph. Okay, thank you all. Thank you all. Now we are moving to the discussion part. The participants, please put your questions in the chat box, or you can directly ask questions to the resource person. Welcome to all to the discussion session. Some questions are here in the chat box, sir. Shall I ask?
Yeah, sure.
Is there any effect of temperature, pH, and light on the color change in flowers and nutritional value?
Of which crop?
They didn't mention that crop.
Okay. Yes. Yes, it is known the temperature has significant effect on the color change. And there is a very interesting study also from Clemson University from a different group, and they published recently in, I think, New Phytologist. So that deeply describe color change in petals, effect on the insect pollinators of this change also.
Okay, sir. Could you explain the use of nano fertilizers for sustainable crop production?
Oh, sure. Yes, the nanofertilizers are super interesting because, as you have seen, these nanoparticles could be introduced, or carry these substances into the plant very, very efficiently. So these can be actually, these nanofertilizers get onto the surface and can be absorbed more efficiently than the other chemicals, because right now we are using substances which can actually allow these things to spread onto the surface. And these substances are called surfactants. And these surfactants are kind of the soap suspensions, and they allow spreading of this, so that the things does not wipe out from the thing. And the other sort of nanofertilizers that are being used are actually people coat the acrylamide beads with these, and they slowly release these into the plants. And also there were examples of the bio clay, where the people have modified the clay, and so you can spray the clay, which is attached either with these pesticides or with the fertilizers.
Okay, sir. Does climate change affect the nutritional value of crops or vegetables or fruits?
Yes, it does. It is quite known from some time, it actually affect both the protein profile and the carbohydrate composition. And I already showed the lipid composition is significantly changed with the heat. As I showed in the soybean example, you can see the lipid composition of the seeds and leaves and anthers is all changed with the heat. So lipids are very important, as we were all talking about the omega-3 fatty acid composition. It actually increase or decrease based on the amount of the heat and drought. And interestingly, we identified like the heat and drought have opposite effect. So the heat reduce the level of unsaturated fatty acids and drought increase it. So if you have like, I mean, a genotype which have higher level of unsaturated fatty acids, it can survive the drought better. But usually drought and heat happens together. So it's a interesting scenario, like, yeah.
Thank you, sir. Any more questions? Any more questions?
I mean, if they have questions related to my research, they can also always directly email me. Like, there's no problem. I will be able to respond back to them.
Okay, sir. Okay, okay. Okay, I think no more questions. So I invite Sandra Yessen, student coordinator of this international webinar, to propose the vote of thanks. Before that vote of thanks, one question is there, sir. BT crops made by your team, is it available in market, sir?
Yeah, we have not developed the BT, but the BTs are available in market in several different crops, in cotton, soybean, and corn.
Okay, okay, sir. Okay, Sandra, please come.
Okay. Good evening to one and all. I deem it a great honor to propose the vote of thanks to all who helped us in making this webinar such a resounding success. First of all, I would like to thank our resource person, Dr. Sachin Rustgi, Assistant Professor of Molecular Breeding, Department of Plant and Environmental Science, Clemson University, who spared the time from his busy schedule to grace this webinar with extremely relevant address on novel strategies to develop crops and management strategies to meet the growing demand for food and fiber. It is reported that more than 2 million US people suffer from celiac disease, which is an autoimmune disease. And now it is a relief that you and your team has created genetically distinct variety of wheat that is safer for people with celiac disease. Thank you so much, sir, for giving us an idea about breeding for sustainable crops production. Thank you so much for such an informative session. We have been fortunate to be assisted by our principal, Dr. Sister Bina Jose. You have been a constant support and have given us a lot of encouragement. Thank you so much, sister. Next, I thank our Vice Principal, Dr. Sister Namida and Dr. Minimor ma'am for their enthusiastic support and guidance. I thank our well-wishers, Dr. Hani Sebastian ma'am, HOD of Zoology, and Dr. Shijati Taragan ma'am, HOD of Botany, for their stewardship, support, mission, and commitment. Events like this cannot happen overnight. The wheel starts rolling weeks ago. It requires planning and a bird's eye for detail. I take this opportunity to thank our teacher coordinators, Dr. Sylvie Matthew and Dr. Honey Sebastian ma'am, and also the organizing committee, Dr. Shijati Taragan ma'am, Smita P.S. ma'am, Dr. Manchu Madhavan ma'am, Dr. Patricia Joseph ma'am, Dr. Fiba Rani John ma'am, Dr. Shiba P. ma'am, and Sister Freddy Jacob. I thank the student coordinators, Hira Babu, Helena Nixon, and Anjali Rajendran, who have been running to make this webinar such a resounding success. We are really grateful to be funded by TBT College Scheme, Department of Biotechnology, Ministry of Science and Technology, Government of India. Finally, I thank the wonderful participants who have turned up in such a great number from various departments. Thank you so much for your cooperation. Once again, I thank each and every one of you and wishing a wonderful day ahead. Thank you.
Thank you all once again. Thank you, sir. Thank you all. Thank you.
Thank
Shall
you so
we
much.
end the meeting? Thank you, sir. All of them can leave the meeting. Thank you.